to the second installment of the structure of immunoglobulin series. Here we will be discussing about paratopes and epitopes. Starting with the paratope. As already mentioned in my previous video, a typical antibody contains a variable region and a relatively constant region. Within the variable region, there are certain special sequences of amino acids which are highly specialized in recognizing the antigen. Such unique sequences of amino acids which are present within the variable region of both the light and heavy chain are regarded as the CDRs. Now it is to be noted that CDRs which are present alone in the light chain or the CDRs which are present alone in the heavy chain are not specialized in recognizing the antigen or incoming pathogen. Or in other words, if I have to put it, then I will put it in this way that the CDR1, CDR2, CDR3 present in the variable region of the light chain cannot detect an incoming pathogen. Similarly, the CDR1, CDR2, CDR3 alone independently cannot recognize an incoming pathogen. That is, to recognize an incoming pathogen and to form the antigen binding site, the CDRs present in both the light and heavy chain of one side of the antibody have to work collaboratively. That is, the CDRs which are present in the variable region of the light chain and the CDRs which are present in the variable region of the heavy chain must work in sync together to form the antigen binding site where they will interact with the incoming pathogen. Now such antigen binding site formed by the CDRs present in both the heavy and light chain spontaneously are known as paratopes. Now coming to epitopes. Epitopes are also known as antigenic determinants. That is, there are certain regions present within the antigen that can trigger an immune response followed by the activation of immune cells and molecules that will lead to the neutralization of the antigen. Now such region which are present within the antigen and that can trigger an immune response within the system is known as epitope. It is to be noted that antibodies too can play the role of an epitope because there are also certain regions within the antibodies which, when inserted within the another member of the same species, may trigger an immune response. Say for example, if I take an antibody from my own and insert it on the body or within the body of some other person or some other individual of the same Homo sapiens species, then my antibody may trigger an immune response in the bloodstream of that other individual within whom I have inserted that particular antibody. So therefore, since such regions are present within the antibodies that can trigger an immune response, therefore antibodies can also act as epitopes. Now here we have learned that both the antigen as well as the antibody can act as epitopes. Having learned that, we need to understand that epitopes can be classified into three different subtypes: isotype, allotype and idiotype. Now what is isotype? For explaining this, I will give you one example. If suppose I take an antibody from my bloodstream and I insert it in the bloodstream of another individual of the same species, and if there are certain regions that do not trigger an immune response within that particular individual's body, then that particular individual's immune system will not be triggered, and therefore the immune cells of that particular body of that particular individual will not be activated. That is, there are certain regions within the antibody which does not trigger an immune response. Therefore, doesn't matter if I take an antibody from my own body and insert it into another individual's body of the same species, the antibody would be treated as the same, and thereby such antibody region are known as isotype. Talking about the allotypes, which is here, Now, there are certain regions within the antibodies which may trigger an immune response. Such regions are known as allotype. On the other hand, the next classification is idiotype, where we need to understand that the CDs present within the variable region of the antibodies also may trigger an immune response when inserted within another individual's body, because these CDs are highly specific and unique. And if that CDs or the framework regions which is between the CDs triggered an immune response, then such CDs or the framework region would be regarded as the idiotype. So what we have understood here, that there are certain regions either within the antigen or within the antibody that may trigger an immune response followed by activation of immune cells, which are known as epitopes. Epitopes can be of three different types: isotype, allotype, and idiotype. Isotype are such regions that does not induce an immune response. Allotypes are such regions, be it present in the antibody or in the antigen, that does trigger an immune response. And the last but not the least is the idiotype, when the CDs and the framework regions between the CDs induce an immune response followed by the activation of different immune cells. So that's all for today. Hope you've understood paratope and epitope. And at the last but not the least, if you have liked the content that is presented in this video, please do like, share, and do subscribe.